Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is part one of an oncology series presented by Clario entitled Revolutionizing Oncology Clinical Trials, the Transformative Power and Promise of AI-Enhanced Medical Imaging. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you are involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading health research and industry company that generates the richest clinical evidence in the industry. Across decentralized, hybrid, and site-based clinical trials, Clario's deep scientific expertise, global scale, and the broadest endpoint technology platform allow their partners to transform lives. And now I'd like to introduce our speakers for today's event. Joining us today are Kim Nguyen, who has 15 years of healthcare experience, supported by his data science master's from UC San Diego. His academic journey included groundbreaking Alzheimer's imaging research using electron microscopy and collaborating with a dedicated team during his graduate program. In his current role, Kim is the forefront of utilizing the latest machine learning techniques to create advanced predictive models, harnessing the power of data to provide innovative solutions that drive results for Clario. Also, Dr. Joel Scherer earned his PhD in 2008, specializing in cardiac imaging. After completing a postdoc in motion management for radiation therapy, he joined Clario in 2011. At Clario, he's been working on image processing software for various medical applications. In 2017, Joel introduced deep learning to the company's projects and currently leads Clario's medical imaging processing group from his base in Switzerland. And Alex Boudreau is a distinguished leader in the field of artificial intelligence with an academic foundation in physics, cognitive sciences, and computer science. With over 20 years experience, he has consistently spearheaded the development of cutting-edge software systems and algorithms. His expertise spans a wide spectrum from pioneering advancements in medical imaging analysis software to leading the development of enterprise speech systems, conversational AI, and cloud-based fraud detection. Now, before we hear from our speakers, I just want to direct the audience's attention to Clario's AI responsible use principles. Note that Clario is committed to employing AI in a responsible way, governed by the highest ethical principles. Additional details can be found at clario.com/solutions/AI. But now, without further ado, I'd like to hand the mic to our first speaker for today. Kim Nguyen, you may begin when you're ready.
Thanks so much, Ryan. So good morning, everyone. Good afternoon, good evening. So my name is Kim Nguyen, and as Ryan noted, I'm joined by my colleagues Joel Scherer as well as Alex Boudreau. Next slide. So for today's agenda, we have this split into three separate sections. I'll be going through predictive progression events within a clinical trial. Joel will then walk us through how we can leverage automation for oncology trials using deep learning. And then Alex will top us off by showing us what next and exciting innovative applications that can be leveraged using generative AI. Next slide, please. So for the first use case, I want to highlight a product that we developed to help accelerate the clinical trial timelines by predicting progressive events within an oncology RECIST 1.1 study using machine learning. What I really want to showcase here is how utilizing over 100,000 anonymized RECIST analysis data points, we can accurately predict progressive-free survival across a patient population. One thing that I do want to note here is that this tool, it's not meant to provide predictive outcomes to single specific patients, but more so to give probabilistic likelihoods of overall progression events across the study patient population. Next slide, please. So to set the stage, I want to give a brief overview of the overall purpose of an oncology trial. So these trials, they're designed to measure the time it takes for patients to experience a progressive event, which in this case, a progressive event is the worsening of a disease state. These trials are very crucial when evaluating the effectiveness of these new cancer trials. One of the key aspects of an oncology trial is what's called event counting, which involves recruiting a sufficient number of patients to experience a statistically significant number of progression events. This process, it's lengthy, it's time consuming, it can often take several years, if not more than that. Next slide, please. So why would machine learning be helpful when it comes to predicting progressive disease? So citing a 2013 paper that you can see here in terms of a spider plot that's shown is this is looking at one specific feature within a RECIST reading. In this case, it's the change of target lesion percent across multiple weeks within a patient population. So if you look at the first couple weeks, there's not too much of a difference between the different classes, but as you move into weeks 5, you move into weeks 10, you start to see a fairly clear and natural divergence within that patient class or patient populations. Red being those patients that do progress, green being those patients that have not progressed. And so this is just using one specific feature, but what if you can actually use 10 features, 15 features, 35 features? Can you take that within some combination in order to make a better predictive result given these features? This is really the power of machine learning. Take n number of features, do some intricate computation and complex relationships in relation to that target variable, and then actually get just a much more fine-tuned and granular result based off of that. Next slide, please. So how does predicting progressive disease benefit an overall trial? So I want to walk us through in the next couple slides is an example mock study design. And if you're designing a study and looking at event counting, you'd know that the number of events that you need to meet your endpoint criteria, taking that particular number, you want to know, then you can actually calculate how many patients you would need in order to enroll to meet your criteria. So I'm just showing this within a formula representation here. So total events at the top divided by the percent of likelihood of a particular patient to progress. And then that reflects how many patients that you would need to enroll within that trial. Next slide, please. So mocking up this example, here's a representation where if you need 100 total events, you know that the expected baseline number of patients to progress is 50%, then given that you would need 200 total patients at the start within a given study. Next slide, please. So if we drill down a little deeper once the trial starts within this mockup, you might expect trial enrollment to look a little something like this. Traditionally, enrollment, you don't typically enroll all your patients at the beginning, but it's more of a gradual pattern, as you can see here. So, for example, a trial might enroll 50 patients in the first 6 months, 100 patients by the end of the first year, and then additionally 50 patients within that final year. And then that might convert into what you see here, where there's 25 events within that first period, 50 events in that middle, and then subsequently by the end of it, you might have that 100 total progressive events that you need from that point. Next slide. But what if we can actually get a better idea of this? Instead of using this 50% expected baseline progression, is there a way that we can actually utilize machine learning to predict of those specific patients specific to the patient population here, if that rate is higher, lower in that instance, so that we can better inform and make study decisions as we're actually moving forward within the enrollment stage. So utilizing that within this example, what if based on some confidence threshold, we can say that the patient population is more likely to have 37 events as opposed to the 25 at that point? What if it could add 75 and by N, 120? that would really give us a sense if we can actually increase or uh, up, increase the pace of the enrollment rate or decrease it, just really depending on what we're actually seeing within this specific patient population. Next slide, please. So looking at this at month 12, let's say we have 75 patients that have already progressed, and we can predict of those remaining enrolled patients, we expect based on that prediction that there's 30 left over to likely progress from that given point. Within this example, instead of actually enrolling 200 patients to start, you would then only enroll 125 within this particular mock example. That's really the crux of what this is intending to do. Can you accelerate your enrollment? Should you decrease your enrollment? Really depending on your specific patient population. Next slide, please. So to pivot from that point, for the next several slides, I want to delve a little deeper into the prediction model setup as well as the results. So for the hypothesis here, as noted before, this is to predict patient progression using the baseline visit and as well as the first and second follow-up visit. The data that we used here is central imaging data that's one, been standardized, two, been anonymized, and three, been completely integrated within our model pipeline. We trained a tree-based classifier model called XGBoost. These models are really great at handling complex relationships across features. They're great at looking at linear and nonlinear data, especially with discarding irrelevant features during its tree splitting as well as pruning. Our overall workflow is to feature engineer and identify key features that would best provide better predictive scores. We review this with and analyze this with our subject matter experts to look at the outputted results, see if there's any gaps within our model itself, review false positives and false negatives, and then we loop through this iteratively so that we can incrementally get better and better results. Next slide, please. So this is an example of a feature importance graph that shows which features are most important within descending order. I'd say that generally when you think of machine learning, it tends to feel like a black box. You take features, you train the model, then you output predictions from there. But these are one of the a nice clean graph that really looks under the hood. So it allows, as we're doing feature engineering, if we're adding more and more features, is it actually signaling? Is it popping? So that it allows us to understand, should we keep those features? Should we discard those features? Subsequently, it allows our subject matter experts as well as us to get a better sense of what the model is doing or which one it's weighting at a stronger signal as it's making its prediction itself. Next slide. So I want to go through a bit of the model metrics and how it's performed up to date. So what you'll see here at the bottom left is the accuracy score of 84%, the precision at 91, and the recall score of 88%. I want to call out specifically the precision score here as within this specific use case, precision is defined as in instances where the model says progression is there. Is there actually progression? And that's represented within the confusion matrix there, where if you look at the top right quadrant and then the bottom right quadrant, those are instances where our model is predicting precision and our scores based off of that. This was tested off of roughly 700 total patients. Next slide. So before I end, I'd be remiss if I didn't actually look or discuss some of our overall architecture that we created in order to help standardize all of the results across these studies. If you're familiar with data science and machine learning, you'll know that 80% of the battle really is data standardization. Our clinical data automation pipeline looks to enable Claire really to map all these studies into a common set of standards. We then automate the workflow and data cleaning process that enables us to create internal and external metrics and dashboards. do data transfer shares, and of course, for the purposes of this example, allow us to perform analysis and create scalable machine learning models. Next slide, please. So just want to conclude and re-emphasize a couple of the main points noted within the section, one of which is machine learning can really help identify complex patterns in order to really make informed predictions. Two is, as for this use case, machine learning can really help better accelerate the clinical trial timelines. And lastly, machine learning, as noted within this use case, as well as what Joelle and Alex will go through, there's many use cases to really revolutionize and change how we think and how we conduct a clinical trial. So with that, this is my last slide. So I'll hand it over to Joelle so he can go through automating imaging in oncology trials using deep learning. Thanks so much.
Thanks a lot, Kim. Good morning, everyone. So yes, I will talk now about the work we're doing to automate imaging in oncology trials. And as we'll see in the next slide, we want to automate not just one aspect of the clinical trial process, but everything we can, while still leaving humans in control, of course. So we can think of the image processing pipeline for oncology trials as being built of 5 major building blocks. First one is image labeling, identifying the data that we receive from sites. Second one is data preparation, preparing the data for reads. For example, in the context of X-rays, you can think about splitting X-rays or rotating them. Next is quality control, looking for any quality issues within images. And finally, reading and analysis. So for this presentation, we'll focus on labeling, quality control, and reading. So the first step, as we'll see in the next slide, is labeling. Labeling means automatically identifying the images that we receive from the sites. As Kim said previously, the very key aspect of all our work is to correctly classify and acquire clean data sets. Specifically in this context, labeling means assigning to every exam that we receive a number of different descriptive labels, such as modality, CT, anatomy, is it the chest, is it the brain, contrast, laterality, directionality, is it an actual slice, is it the sagittal acquisition? And automatically identifying all these labels enables us to do many things. First of all, it enables us to check protocol adherence by making sure that all the required sequences have been submitted and that they are according to protocol. But it also enables further downstream automations by triggering the correct models depending on the data that is received. This labeling is supported by a large family of different models broken down by anatomy, by therapeutic area. And these models make use both of image data in the forms of CNNs and also DICOM tag information and metadata using tabular models. This is an ongoing work. We are constantly improving the accuracy and adding support for new exam types. We currently support around 200 exam types and have labeled more than 1 million exams so far. Next slide, please. So once the data is labeled, we can move on to quality control. And this means checking for common quality issues. The issues we look for depend on the modality and the application, of course. In the context of oncology, we can look for incomplete coverage. So basically asking the question, is the anatomy complete? Are we missing some slices? Is the field of view correct? Another one is patient verification. It's a rare problem, of course, but it's becoming harder and harder to check for in the context of increased privacy regulations and patient de-identification. So we want to, using the image data only, be able to ensure that the data we receive at follow-up corresponds as the same patient as the patient we had at baseline. We can also look for various artifacts. So together, these models enable us to have a faster turnaround time, because every time a model finds a potential issue, the case can be flagged for expedited review by a human. This in turn leads to higher query success if less time has occurred between the first acquisition and the time the quality problem is detected. There's a higher chance that we'll be able to have a repeat scan or resubmission of the exam. And this in turn leads to higher data quality. Here as well, our models reach good accuracy. For example, we have more than 90% accuracy for our coverage model. Next slide, please. So once we have ensured that the data is correctly labeled and of good quality, we can finally turn to reading and prepare the data for our radiologists to review. So in the context of oncology trials, there are several reading paradigms, but the gold standard is really RECIST for solid tumors. As you may know, RECIST consists in the reader selecting 2 target lesions per organ for a maximum of 5 target lesions at baseline, and at follow-up, measuring the previously chosen lesions, as well as identifying any new lesions. And here our objective is really to help the radiologist as much as possible, save time for them, while still leaving them in control. So in the next slide, we'll see our models for baseline. Here, the goal is basically to detect and segment any lesions that are seen in the CT scans. Detecting and segmenting lesions overall in the entire body is an extremely challenging problem for machine learning, even for the most advanced deep learning models. So we break it down by anatomy to make it more tractable. And we have focused on the most common anatomy so far. Once the lesions are correctly detected and segmented, we can automatically select the lesions that qualify for RECIST, for example, by applying simple size thresholds before we present them to the radiologist. And this is the last and really a key step. It's to have a great UI to show these AI findings to the radiologist and allow them to make use of them easily by facilitating navigation, selection, and even editing or discarding them if needed. And here we have some figures for our segmentation models. We reach 75% accuracy for liver, 85% for lungs, and 81% for lymph nodes. Next slide, please. At follow-up, the problem is a bit different. We have identified the lesions previously. The reader has already read the previous time points. So we're really looking to track these lesions over time. And the process for doing that is to start by predicting the correct slice. This sounds like a simple step, but it saves the reader a lot of time by avoiding them having to scroll through the entire volume to reach the lesion. Then we can automatically locate and delineate these baseline lesions on the follow-up scan. And here we use segmentation as well. We simply detect and segment all lesions that we can on the follow-up exam and use a matching algorithm to see which ones correspond to the baseline scan. Any lesions that are not accounted for by this matching algorithm are assumed to be new lesions and shown as such to the reader. And here again, the key is to provide the good UI for the reader to automatically jump to these predictions and accept them or make edits as required. And here our slice prediction accuracy is above 98%. Next slide, please. So RECIST is the gold standard and will probably remain the gold standard for many years to come. But having AI models provide 3D segmentations actually enables us to go further than RECIST. With the volume, we can look at more precise and maybe more sensitive measurements of disease, such as total tumor burden or texture analysis of the lesions, lesion classification, which in turn allows to go to more advanced models of disease, such as tumor growth dynamic or full-blown radiomics. So in conclusion, AI for oncology trials enables us to improve our efficiency, improve our quality by decreasing our turnaround time, detecting quality problems, and reducing reader variability. And with that, I'll hand it over to Alex to tell us about generative AI.
Thank you, Joel. And that's a great segue to this last topic, which is the applications of generative AI in clinical trials. Next slide, please. So you might be asking yourself, does generative AI actually apply in clinical trials, right? And when you think about the definition in simple terms, which is a system capable of generating novel content with, which is text, audio, or images, for example, the question is even more relevant. However, it's important to note that generative AI has evolved well beyond synthetic data generation, such as applications of neural style transfer or deepfakes, right? Like voice cloning, for example. And what we're seeing is semantic understanding, basic reasoning, and problem solving emerging as capabilities from Gen AI. And more recently, when we think about that topic, a lot of people will refer to ChatGPT. And what's interesting with ChatGPT is that's a conversational AI application that leverages the GPT family of models, right, which are large language models. And LLMs are in fact a type of Gen AI as well that is specialized in natural language processing tasks. And these models have become very, very good at understanding and generating natural language. And what we're seeing now is that users are able to hold complex conversations, summarize information with these models, translate languages, write code, and so on. And the list of applications goes on. And what we're seeing is really generative AI exposes capabilities that is far beyond next word prediction as initially intended. Which leads us to the next topic. Foundation models, right? What we're seeing, and something that is very, very interesting in the realm of generative AI, is the concept of foundation models, which are large pre-trained models that exhibit a broad set of capabilities, but that can be fine-tuned or specialized or taught new tasks, unseen tasks, with prompts and fine-tuning capabilities. And what's interesting is that LLMs have very large neural architectures, and that's why they're called large language models, right? They have billions and hundreds of billions of neural network parameters, and they're pre-trained on a vast amount of data comprising of billions and trillions of tokens. And in this context, a token is could be a word or a grouping of characters, right? And those are extracted from books, articles, source code, conversations, scientific papers, and the list goes on. And it's that large pre-training that actually gives the power of those foundation models and that opens up capabilities never before seen. Next slide, thank you. And which is why it's really interesting, because those LLMs now can be used in the concept and context of clinical trials, right? We can use those technologies to interact with clinical data, such as study protocols. Now, LLMs are for text processing, right? So we're looking at documents here. So what we can do with these models is that we can ask specific questions about a study. We can ask, for example, where are the measurable lesions? What is the defined response criteria for that study, and other topics as well. We can also then use the capabilities of summarization and extraction for creating training material for the staff that is participating in the study. Or in another application, we could, for example, find similar studies based on the semantic similarity of one study versus past studies, using attributes of treatment, disease indication, and other attributes that are defined within the study. So that opens up quite a few capabilities here. And this can be achieved with a concept or a method called retrieval augmented generation, right, and commonly known as RAG. And that has become very popular in the field, and you'll hear a lot about it, and for good reasons, right? It's a very powerful technique. And this leverages methods such as semantic search and prompt engineering. And how it works at high level is that you take a document, separate it in chunks or segments, and then you send those segments to what we call an embedding model, which essentially extracts a semantic vector representation from that content. Those vectors are then stored in a database, such as a vector database, right? And then when a user asks a question pertaining to your document, that question is also embedded to create a semantic vector. And we're looking for the most closely related information and content using semantic search. And that information can then be inserted into the LLM as context for generating an answer specific to the question and specific to the document at hand. If we move on to the next slide. This is an approach that is proving to be extremely powerful and versatile, but it's important to note, especially in the context of clinical trials, that's not without risk, right? What can happen is that sometimes you can have an answer that is incorrect, that is incomplete, or that may also include what we call hallucinations. And hallucinations in the scope of LLMs is an answer that sounds very good, that sounds accurate, but is it actually not grounded in facts, right? So it's incorrect. It just looks very nice and it's well formulated. So that can be a little bit dangerous. But fortunately, there's ways to mitigate those risks, right? There's strategies to mitigate those risks. And what we're using here at Clario is a multi-layered AI approach where we augment the standard RAG process. And how it works at high level is that you take the document, you break it down into segments, and instead of directly submitting that for embedding, you do a first step of classification. So you use another AI model that looks at the chunk of information, determines if it's high quality content, and if it's relevant for the application at hand. So we can filter out invalid information or information that does not add value. Once you've got that classification done, you can go ahead and proceed with the embedding to retrieve the semantic vectors. And then when the user asks a question, we do the semantic search process. But instead of providing the answer directly to the user, we use a secondary AI model that does essentially a validation on the answer. It looks at if the answer is of quality and semantically related to the content and the question. You can actually have a scoring mechanism that determines, okay, is the answer good enough for me to present it to the user? So you can consider that as a AI judge on the side that really helps augment this process and provides more accurate and higher quality answers. So this is proving to be quite effective. And that whole RAG process is evolving as more techniques are refined. Next slide, please. Now, when you think of clinical trials, of course, there's more data than text, right? It goes beyond text. We have images, we have biomarkers. And in a recent study, it was found that 67% of oncology trials that were registered in clinicaltrial.gov actually relied on imaging for at least one of their endpoints. And that shows the importance of imaging in oncology trials overall, right? And interestingly, what we're seeing in the industry is with the success of LLMs, right, of generative AI with large language models, similar methods are actually adapted to leverage for other modalities. So we're looking at the power of the LLM architectures and using that to create other types of foundation models. And we're seeing an emergence of various models, in fact, that tackle vision, that are multimodal LLMs, such as OpenAI's GPT-4v, that offer also promptable image segmentation, such as Meta's segment anything model. If we take that as an example, you have the diagram on the right here, that illustrates a high level view of how SEM works, right? So you have an image, And then you have an image encoder that actually extracts the embeddings, right? So the semantic representation of that image in a vector form. Then you also have a prompt encoder, which is fed a prompt, which could be a bounding box, a region of interest, a point, or textual description of what you want to segment, right? You feed that to the encoder. Jointly, those embeddings are fed then to the max decoder. And what this does is actually selects the optimal final segmentation mask based on the image and the prompt. And this is proving to be actually quite powerful. And what's interesting is that the segment anything model has been pre-trained on a vast amount of natural images, right? Think of photographs and scenes and so on. So it is, in fact, a segmentation foundation model. Next slide, please. And what's interesting as well is that we're seeing expanding capabilities that go beyond just natural images. A study that was conducted at Duke University actually used the segment anything model, and it evaluated its out-of-the-box segmentation performance on 19 medical data sets that span various modalities and anatomical regions. And what they found was really exciting to some extent, right? You look at the results here. In one side, you see a low accuracy that it achieved on spine MRI images. However, if you look at the hip X-ray results, it achieved a much higher accuracy score for segmenting that at specific anatomical region. And what we're essentially finding from that is that a foundation model that's not even been trained on medical images is actually capable of segmenting medical images in some degree with fairly high accuracy. Next slide, please. So this shows really the power and the opportunities for developing multimodal medical foundation models, right? So you think about it, there's now a possibility, an opportunity to train foundation models that span imaging modalities and anatomical regions. And what we can do is create a single model, quote unquote, expert model that can do image understanding, segmentation, interpretation, and which leads us, as Joel mentioned previously, to the concept of total tumor burden analysis, right? That can also include radiomics, and that can provide as the output a confirmation of response, for instance. And what this does really is opens up the reality of having an AI companion in partnership with a radiologist, for example, where you can feed in the multimodal aspect, you can feed a protocol, a study protocol, a series of images, and then you have the model that actually goes and interprets automatically the study protocol, performs automated image analysis and segmentation, and can even provide an explanation of its analysis in natural language to the expert radiologist. So that opens up a brand new realm of capabilities in the clinical trial space and medical space in general and overall. Next slide, please. So what we've seen is that generative AI and AI in general, in fact, is a very rapidly evolving field where you're seeing increasing capabilities, right? If you think about foundation models in language, such as large language models, we're seeing an evolution towards vision and now multimodality and other modalities in the future, right? What this opens up, though, is that it requires further considerations on privacy, on safety, reliability, and accuracy. That's why keeping humans in the loop is extremely important, and I think even more so, especially with the concept of generative AI. But what we also need to make this possible is to perform pre-training at scale with medical data. Now, the question is, how do we go and obtain that data, those data sets and that data that is of high quality and accuracy such that we train a model that is highly capable but also very accurate. And I foresee that this is going to require many collaborations that spanned companies and organizations in the future, such that we have really highly capable generative AI models for the medical and clinical space. So as a concluding point, just to demonstrate a little bit where we are in the industry in that space, I fed an image to, sorry, if we go to the previous slide. Yes, so what I did is I took an MRI scan, public domain, that includes a lesion. And I fed that to OpenAI's GPT-4V model and asked it to interpret the image without any details. And what you see here in quote below is a sample, a portion of that response that confirms it's a T2 weighted MRI image and that it includes a lesion that could be, for instance, a glioma. Now, this is really interesting, and I'm going to leave it at that and hand it over back to Ryan so we can open up for questions. Thank you very much.
Well, thank you very much for that very insightful presentation. I would like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions from the audience, so we'll get ourselves started with those. Very first question I have here from the audience simply wants to know, what are some examples of features used for predicting the number of events for a participant? Sorry, Kim, I think you might be muted there.
You're right, I'm muted. Yeah, that's a great question. I could, so two different, well, one that was, I think when we gave it to our subject matter expert, which was a little bit more what they expected to find, is a lot of response diameters that were referenced specifically within the resist reading itself. And one that was actually fairly surprising that popped and signaled fairly high was just how long a particular patient was on study itself. I think that was one of the more surprising ones when we provided to our medical affairs group, because there wasn't inherently a thought that there would be just as strong of a correlation with whether or not a patient would progress or not progress just simply by how long they were on the trial itself, or it would have such a strong influence of how the model would react. But those were two groups or classes of features that were fairly interesting when we were reviewing feature importance.
That's great. Thank you so much. The next question here is curious about what are some of the challenges when training a model that identifies imaging modality and anatomy?
Yeah, thanks, Ryan. Well, modality isn't much of a problem. The DICOM standard tells you what modality the exam is, and that's usually fairly reliable. Anatomy is a much bigger challenge, of course. If you think about it, some anatomies might be easy to predict, like is it the brain, but you need to support that for different types of sequence, for T2, T1, CT, X-rays, sagittal orientation, axial orientation. Some anatomies might be more challenging or lead to confusions, such as hands and feet, for example, or have ambiguous definitions, like is your exam a chest and abdomen exam with insufficient coverage, or is it just a chest exam? That can be hard to tell. So the solutions to these problems are, I think, the fundamentals. You need to have a good data set, a diverse data set, but also balanced data set, and of course an accurate and clean data set. And with this in place, I think 90% of the job is done. Of course, there's also some machine learning tricks to get better results, such as using data augmentations, but really the fundamentals are what matters.
That's great. Thank you very much for your thoughts there. Alex, I've got a question I think is directed to you, and I would like to know just about what you think is an aspect of generative AI that makes it promising for medical image analysis.
That's a really good question. Thanks, Ryan. One aspect I found really interesting with LLMs that actually can be translated to imaging is the capability of zero-shot to few-shot learning. And what that means is that with zero-shot, you provide, let's say, a question to the model and provides a good answer. and it knows how to do it, even though it's not been trained for that task. And with few shot, if that zero shot doesn't work, you can give it a few examples. You can give that model a few examples to show what you're expecting as an answer. Then it will be able to answer accurately follow-up questions, right? So that's actually really powerful. You don't have to retrain the whole model to answer new tasks. And this actually translates really well to the concept of imaging, where you can see a future where the radiologist, for example, gives a segmentation as an example. And the model essentially now learns how to segment that specific lesion accurately for unseen images going forward, right? So that's a very powerful technique where instead of retraining a new model with hundreds of thousands of images, for example, you can have that foundation model that can adapt very quickly by showing it just a few examples. So for me, that's very exciting and very promising. And related to that is the concept of self-supervised learning that we're seeing being very effective in generative AI, where the models learn to understand the patterns in the data by essentially, quote unquote, doing unsupervised training. So there's a lot of techniques there that are very exciting that can be applied to imaging or are being applied to imaging as well.
Wonderful. Thank you very much for sharing your insight on that one. I have a question here regarding predictive progressive disease. The question is wondering, how can we rely on the results of predicted PD, considering that each patient may respond to any given investigational drug in diverse ways? Could you please explain further, perhaps how reliable the results of predictive PD are?
Yeah, this is a really great question, and one that we actually had a lot of internal discussions about. One of which is, do we want to provide specific results at a patient level, which I think we're at a point where, given how new this particular model is, I don't think we're comfortable enough to say, for a given patient, let's provide a result to the sponsors for that specific patient. That's why we really want to focus this more so on the overall patient population, and based on that specific threshold, can we say something more about the overall population as opposed to one specific patient? As the question noted, there can we actually get to the point from a patient safety standpoint, say that this patient is A or B, given what actions can occur from that given point. So we really try to focus this more so on an overall class of the population, as well as just more so from just a statistical standpoint. Can we actually say something on that more generic level?
All right, very good, thank you. The next question is curious if you're able to measure the tumor volume three-dimensionally, and do you think that this would be a much better representation of tumors in reality?
So the answer is yes. AI models give us a 3D segmentation out of the box, and we actually reduce that to 2D to fit with the RECIST protocol. Now, of course, I want to stress that the accuracy of this 3D segmentation completely depends on the anatomy and the type of lesion. But for the most part, and for the most common locations, I can say that the 3D segmentation is accurate and I think would be useful. Now, there is some debate, I think, within the community regarding the usefulness of 3D volume measurements for tumor representation. It is more informative than just the diameter, but RECIST has shown its usefulness even with a single diameter. So I think more than just the 3D shape of the tumor, the important thing is that it enables us to go further by, for example, using multimodal imaging.
Wonderful. Thank you very much for the answer to that question and for all of the answers today. But we have reached the end of our Q&A portion for the webinar. As a reminder to everyone that this is part one of a series. There's more in this oncology series by Clario that you have already been registered for. Part two is using scientific validation to move the needle on accessibility functionalities, which is scheduled for Monday, December 4th, and then again come back on Wednesday, December 13th for integrating objectively measured safety endpoints and patient-reported outcomes in oncology trials. Now for today, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, there is a link to view the recording of this event, which I've put into the chat box, which you can share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, and have a great day.